Hey, what's going on? It's Mr. Salmingo. This is the last video of AP Biology, so I hope you pay attention, hope you understand. In this particular section, we'll be going over the math portion, or even there's even math portion in the free response. So I just want to go over the formula sheet that they give you and 6 sample questions of the fill-in part of the AP exam. So as you see in front of you, this is the worksheet you're going to get at the beginning of your AP exam. So you get all these formulas, you get all this stuff. You don't need to memorize it, so you just need to know how to use it. Some of these we've never done, and some of these you should have learned from your old math classes, so you may have forgotten. But I'll go over it very briefly and very quickly. I know you guys don't like long videos. First of all, there's four things they're going to ask of you, or possible things they're going to ask of you. They might ask you about standard error. They might ask you about find the mean of something, or standard deviation, or chi-square, and chi-square comes up a lot, and most of you haven't seen chi-square, so I'll go over that really quickly. For standard error, basically when you do a lot of experiments, there is a variation of what you're supposed to get or what you don't get. The standard error, how you calculate it, is you take the standard deviation, S, and divide by the square root of n. And before I forget, on your exam, I never told you guys this, and I'll tell you guys again over Schoology, but you're allowed to use a calculator. You need to bring a calculator. Bring a calculator. All right. To find the standard error, you get the standard deviation, which is here, and divide it by the square root of n. N represents how many trials that you run. So for example, if you did your experiment 3 times, or 4 times, or 5 times, you calculate the standard deviation and divide by the square root of n. There is some, on the free response, there is partial credit. On the grid-in, on the multiple choice where you do the math, there is no partial credit, or getting credits for just putting stuff down. But in the free response, there is. So if they ever ask you for anything, or ask you for one of these, just write down the formula and try plugging in numbers, and you'll get some partial credit. Standard deviation looks like this. Some of you in calculus know what this means, and some of you don't know what this means. This sigma means the sum, means to add all the add everything, basically. So to calculate standard deviation, you add xi, x meaning the, and I meaning like you're basically adding every value that you have, minus x with a bar on top of it. And you may be wondering, what's x with a bar on top of it? X with a bar of it is the mean. And again, you don't have to memorize this. This is all in your worksheet. You just need to know how to analyze and understand what to plug in. Remember, this is not a math test. They just want you to know where to plug in into which formula. And then you divide that by n minus 1. Again, n, as I told you before, is how many trials of the experiment did you do. And that's how you calculate standard deviation. The mean, you guys know how to calculate the mean. This is a simple way of calculating them, or a complex way to calculate the mean. The formula is this formula is ugly. But basically, this piece means, let me erase that. This piece means add up everything, add all the data that you have, and then divide by how many you have. It's just like calculating a batting average, like calculating your grades. Add up all your test scores and divide by how many tests you get. That's how you get the average, or that's how you get the mean. And the chi-square, this is the one I'm going to go over with you today. And in case you forget about this, or what these things mean, ta-da, there's on the side of your sheet, they have every like a legend for all of what the letters mean. So this particular thing means chi-square. Chi is a Greek letter. O stands for the observed, and they give you the observed. And E is expected. So what were you supposed to get? And O is what you actually got. So when you do like genetics, you're expecting to get, say for example, half of them are supposed to be black, but 75% of them came out to be black. So you use chi-square as kind of a statistical analysis of genetics. And again, in case you forget anything, if you look over here, S is the sample standard deviation, X with a bar on top of it means mean, N is the size of the sample, or how many you have, O is observed, E is expected. And then this is very important because we never went over this. Degrees of freedom is the number of distinct possible outcomes minus 1. So you're saying, what the heck does that mean? So on to the next piece. Here's a chi-square table. There is a possibility that on your free response or on your math section, they're going to say, what is the chi-square, or something like that. This is what they have to give you. First of all, you calculate the chi-square, which is good. And then they're going to tell you a p-value. They have to tell you this. Whether the p is 0.05, they're going to tell you the p is 0.01. You don't need to know what p is. It's just going to confuse you. But just know in the question they have to tell you, calculate with p as 0.05 or calculate p as 0.01. If they give you 0.05, you're going to use this top row. Okay. If they say p is 0.01, you'll use the bottom row. Okay. Now, this thing that I boxed, it says the degrees of freedom equals the number of possible outcomes minus 1. So if there are 4 possible outcomes, then you'll be looking at, the answer you're going to give is 7.82 or 11.34, depending on the p they give you. And for the most part, they really stick to like these lower numbers. They don't really use the bigger numbers. But I'll go for show you some examples. But if there's only 2 possible outcomes, or if there's only 2, like there's only capital P, and lowercase p, there's only 2 possible outcomes, that means you're going to use the 1 degree of freedom. And that's how you read the table. Again, you don't have to memorize it, you just have to know how to read the table. This part, this big half, this top half, is really the hardest part of the formula sheet. Everything else, I think you guys understand. Everything you guys and girls understand. Here on the right, you have all the prefixes, so in case you're confused what they mean, giga is 10 to the 9, kilo 10 to the 3, micro 10 to the negative 6, and here's the symbols for the metric, just in case you're confused. Laws of probability, we went over that very briefly. If they're mutually exclusive, like you're trying to calculate the probability of something happening, you add the possibilities. But if they're independent, you multiply. Okay. Hardy-Weinberg, we're going to go over this again today with some sample questions, but you should know these formulas. They're given to you, but you should know what they mean. You should know P means the frequency of the dominant, and Q represents the recessive. And some of you may be wondering, how come there's 2 formulas? Remember, P squared represents the double dominant, 2PQ represents the heterozygous, and Q squared represents the double recessive. So that's what that means. And P and Q represents just the frequency, P meaning dominant, Q meaning recessive. Okay? Mean, median, mode, you should all know that, and you should also know the range. You know, range is like the biggest minus the smallest. Like how wide is that range? You should all know that from math. And the second page of your formula, I don't even go over this with my AP Bio kids just because they have good headings for you. So if they're asking for something, you can just look at the formula sheets on this page and find it. So if the problem says, hey, look at population growth, there it is. If it says exponential growth, there it is. If that's for logistic growth, there it is. Like they give you the formulas. The one thing that you might not be familiar with is water potential, which is right here. Okay. The water potential is equal to the pressure plus solutes. They usually give you, they might give you this, and you have to solve for the S, or they give you P and S, and you have to solve for the general water potential. But it's a very simple formula. If I want to know the water potential, you have to add the pressure plus the solute. And then the solute potential of the solution, again, look for these words. If you don't understand, look in your formula sheet. Here's the formula to find out the solute potential. Negative I CRT. And you're like, what the heck is negative I CRT? Here's what I CRT means. It's right below. The ionization constant, they usually give that to you. The molar concentration, which they give to you. The pressure constant, which is 0.0831. And then the temperature, which is 273 plus whatever degrees in Celsius. Again, the formulas are all given to you. You don't have to memorize this. Same thing with the temperature coefficient. Same thing with primary productivity. Again, they're going to ask you for it. All you have to do is plug it in. In temperature coefficients, we have these Ks. Again, they're going to give you those Ks. And T represents time. Okay. Primary calculation, if they're looking for mlO2 per liter, you know you have to multiply by 0.698. If they ask you to calculate carbon fixed per liter, you know you're going to multiply by 0.536. So look into the key words of the math questions, look in your formulas, and it has to be there. Oh, sorry, this T is not for time. This T is for higher temperature and the lower temperature. Okay. See, it's even nice. Not only do they give you the formulas, but they tell you what it stands for. So don't be afraid of this formula sheet. Okay. And the last thing is all things that I think is very, very old or stuff that you should already know. So here are the formulas for anything about volume and surface area. Here's what all those letters mean in case you forgot. Here's the Gibbs free. I don't see this very much in the AP Bio test, but if they ask you about Gibbs free energy, there's the formula, and here's what you plug in. You should know pH is measured in a logarithmic scale. We talked about that before. And then dilution, how to dilute something. I means initial and F means final. So give me the C, the concentration and the volume of the initial, and I can tell you what should be in the final. So if they ask you a question about dilution or to calculate something about dilution, you know where to go. So this is your sheet. You get this at the beginning of your test. Again, you need a calculator. Bring a calculator to your AP exam. So let's go over, I just want to go over 6 questions with you all, 6 possible questions from the sample AP exam, and which are grid-in and which are math. So I'm going to pull up, I guess this was 2 years ago, practice exam. Okay, I'm going to go over this possible questions. You might see 1 or 2 of these exactly or very similar to how it is now. Number 1 says, use the graph above to calculate the mean rate of the population growth between days 3 and 5. Okay, very simple. This is like an algebra 1 problem. Okay, so it says, what's the mean between 3 and 5? Mean meaning the average. Well, here's 3 and here's 5. At 3, you have 200, and at 5, you have 900. So to calculate the mean is like calculating the slope. So think of y2 minus y1 over x2 minus x1. y2 is 900 and y1 is 200. The y values for the 2 points. Now on the bottom, we're doing 5 minus 3. Again, you can use a calculator if you want, and you're going to get 350. For these math questions, you don't have to put units because you're just bubbling in a sheet. But if you're doing free response, you have to put units or you get marked off. So that's how you calculate mean on a graph. You'll probably see a question like this. Not so hard. Okay, so that's 350. All right, this one, number 2, is one that you probably have never seen before. You should have, but most of you haven't, so I'm going to tell you how to do it. It says here, certain species of flowering plants, the purple allele P is dominant to yellow. So they did a cross between a purple and a yellow plants. When they planted, 146 were produced. They got 87 purple and they got 59 yellow. Calculate the chi-squared value for the null hypothesis that the purple flower plant was heterozygous. Give your answer to the nearest 10th. So this is the part where you're like, what the heck are they asking for? And this is the part where I tell you, don't be nervous, you got this, okay? First of all, the question says, hey, what's the chi-squared value? So you know I'm gonna go back to my formula sheet. I'll even show you. And here's the formula for chi-square, okay? Sum of O minus E squared over E, okay? So I'll go back to my problem, which is right here. And the formula, again, is the sum of O minus E squared over E. And that equals chi-squared. So that's what I'm trying to calculate, okay? So I have observed and expected, okay? So there are 2 colors, right? There's purple and yellow. So I'm gonna have to, when I say sum, when you see this sum thing, I'm gonna have to do this once for purple and once for yellow. So first of all, it says do it for heterozygous. Okay, so let's pretend. Okay, one of them's supposed to be heterozygous, so I'll do capital P and lowercase p. And the other plant was yellow, so it has to be lowercase p, lowercase p. When I do the Punnett square, you'll notice that half of the kids should be purple and half of the kids should be yellow, but I did not get that. Okay, so let's start off with purple. Okay, let's start off with purple. Now I observed 87 purple flowers. Now, this was 146 seeds. I should have got 73 purple and 73 whites, but instead I got 87 purple and 59 yellow. So for purple, my observed is 87. I'll do a little O. My expected, it was supposed to be 73 because since it's half and half, I should have got 73. Okay, so to calculate the chi-squared for this, for purple, it's 87 minus 73 squared over 73. And to make this easy for you, you should get like a 2 point, I'm just gonna make something up, it should be like 2.5. Okay, so that is, remember, the formula is the sum of it. All I've done is purple. So I've already got one of it, 2.5. Now let's do it for yellow. Yellow, I observed, if I look at my problem, I got 59 yellow flowers. I wasn't supposed to get 59 yellow flowers. I was supposed to get 73. Remember, it's supposed to be half and half. So observed, I got 59. My expected is 73. And now I plug it into the chi-square formula again. 59 minus 73 squared over 73. And if I add this up, I think you get 2.5 again. Something like that, okay? Remember my chi-square formula, which is right here, is sum all of the observed and expected calculations. So for purple, I get 2.5. For yellow, I get 2.5. Your answer is going to be somewhere about 5. Don't quote me on this. It's probably a little bit wrong. You put it into a calculator, but you should get something around 5 as your answer. And that's how you do chi-square tests. Find the observed, find the expected, plug it into the formula. Okay, number 3, it says, and then be careful on this question. It says, what percent of the biomass in the forest community represented above is tied up in the shrub layer? Here's my shrub layer. Give your answer to the nearest whole number. So you notice it's a very complicated chart, but after the sun, the energy that's accumulated is 1.9 10 to the 4th. Or if you don't like scientific notation, I move my decimal point 4 spots. This means the same thing as 19,000. This tree layer, like of all the energy that's coming from the sun, gets 13,000. Okay, again, I just moved the decimal point 4 places. This one, the herb layer, only gets 1,100. So if this whole thing is 19,000, this gets 13,000, this gets 1,100, you know that the shrub layer should get basically these two added and subtract from 19,000. So if I add those together, I get 14,100. And then when I subtract from 19,000, I get 4,900. Yeah? Yeah, 4,900. Okay, the biggest mistake someone will put is like, okay, my answer is 4,900. But that's wrong. The question didn't ask how much is in the shrub layer. The question asks what percent. So the last step that they want you to do is take your 4,900 and divide by the 19,000 that is total in the forest. And I think you get something around 25% if you put it in a calculator. Okay. Okay, number 4. You will get a Hardy-Weinberg question, and I don't think it's that difficult. I'll teach you how to do it. You did it with Mr. Shu and I was gone. It says here that brown skin is dominant for green skin. So I'm going to say brown skin looks like this. This is still brown, but it's heterozygous, and then this is green. And it says there is a drought, and when the drought ends, 12% of the remaining frogs exhibit the green skin phenotype. If the population is in Hardy-Weinberg equilibrium, what is the frequency of the green skin? So remember, you know the key word here is Hardy-Weinberg. You know you're going to be dealing with P's and Q's. Okay. Now, it tells you that 12% have the green skin phenotype. Okay. And the question is, what is the frequency of the green skin allele in the next generation? So 12% of the frogs are green, but what is the frequency of the allele? Or, since green is recessive, they're basically asking, what the heck is Q? Okay. So, to set up the Hardy-Weinberg, I'm going to do the first equation, which is P squared plus 2PQ plus Q squared equals 1. Because 12% of the frogs are green, that means that this Q squared is equal to 0.12, right? And this part right here, the rest of the frogs, the brown frogs, this is 0.88. Okay? But the question doesn't ask for that. The question asks, what is the frequency of the green skin allele? Or again, what the heck is Q? Well, if we know that Q squared is 0.12, to find Q, all you have to do is take the square root of 0.12. And when you do that in your calculator, you should get, I think, 0.35. 0.35. That's the frequency. in the case, just in case, because the AP exam wasn't going to ask the exact same questions, what if the question said, what is the frequency for the brown skin allele? Well, you found that Q is 0.35. The second equation of Hardy-Weinberg is P plus Q equals 1. So if Q is 0.35, then P has to be 0.65. So just in case they ask you that question, you know how to calculate P and Q. You take whatever they gave you, I put it in my formula, like Q squared is 0.12, and I took the square root to find Q. All right, last question. Oh, sorry, 2 more questions, both a little bit easier. All right, this one says, use the graph to calculate the lag time in months between the change in the densities of the prey and the predator. So to find the lag time, what you're gonna do, so here's the population of the prey, and then eventually the predators come, and then the prey goes down, and then the predators goes down. So if I want to find the lag, I need to find a common point, like a peak or a valley for both prey and predator. So for me, what I see that I have a bottom here, and I have a bottom here. So my lag time, you see here, the lag between this bottom and this bottom is about 1.5, right? 1.5 boxes. So my answer is 1.5 months. Again, the math section is not supposed to be difficult. It's not a math test. They just want to know that you know how to calculate the simple things in biology. And the last question, it says, how much carbon is released into the atmosphere as a result of the herbivores? Okay, so first thing I would do in this test is, where are the herbivores? Okay, they're right here. And some of it goes into respiration, but how much? Again, this is not supposed to be difficult. It's not a math test. Well, I notice that 5 of the herbivores goes towards predators. And I notice that 60 goes into the soil. And I know that 125 goes in. So if 125 goes in, and 60 goes out, and 5 goes out, to calculate this, it's basically 125 minus 65, or the answer is 60 has to leave by respiration. So again, to repeat, 125 went in, 60 went out, 5 went out. So 125 minus 65 means this arrow has to be 60. So that is my quick and brief review about the math part of biology. Again, even if you didn't know chi-squared, that's 5 of the questions you should know. And Hardy-Weinberg, I don't think you should be intimidated by Hardy-Weinberg. Again, there's only 2 equations, and they give them to you. So good luck on your AP exam. I have a lot of faith in you. If you want to practice, I'm going to put this practice exam also in your Dropbox. And I'm also going to put some of the old free responses on Schoology. So if you want to look at the old free responses, especially those that have math, you could practice those as well. So take care, enjoy your last video, and I'll see y'all later.